Our next speaker is Professor Dame Pamela Shaw, a neurologist. And Pam's day jobs include Director of the Institute of Translational Neuroscience at Sheffield and Director of the Sheffield BRC. So I don't know when you'd have time to sleep, actually. These are big jobs. But anyway, Pam is going to talk about precision medicine approaches to MND. Thank you. Pam.
Thank you very much.
Right, there you go.
So thank you so much to Munir for inviting me to come and talk today. It's very apt, actually, because this is MND Global Awareness Day today. So it's good to be doing a talk on MND. So what I'm going to cover is the challenge of the heterogeneity of the disease, how genetics can help us subclassify MND, how incorporating experimental medicine into clinical trials enables us to dissect out subgroups, responders and non-responders, and so on. Precision approach to therapy development using human cellular models. We can now take skin cells and reprogram them to become motor neurons or glial cells, and identifying responder subgroups in future trials using biomarkers of therapeutic efficacy, which are emerging now, very exciting. So this disease, we've tended to treat it in clinical trials as though it's one thing. We know it's not. It's heterogeneous clinically, genetically, pathologically, and prognostically. And when we're trying to develop therapies, neuroprotective therapies for this cruel disease, we need to take account of this heterogeneity. So clinically, a lot of patients develop classical ALS, as described by Jean-Martin Charcot, but people present with segmental syndromes involving bulbar, upper limb, lower limb function. Some have progressive disorders of lower motor neurons in the brainstem and spinal cord. Some have a predominantly upper motor neuron problem. And some patients, of course, have concomitant frontotemporal dementia. So it's very genetically heterogeneous. At least 30 and more genes now are known to cause or predispose to MND. The first gene found back in 1993, SOD1, SOD1, not a bad name for a gene that causes the worst disease in medicine. And C9orf72 is the commonest gene, SOD1 is the second commonest. But there are a whole host of other rarer genes that have been found with developments in genetic sequencing. My husband is a neuropathologist, so I always have to show this slide. But even pathologically, it is heterogeneous. So 97% of patients with MND will have what's called a TDP43 proteinopathy. So TDP43 is a protein that normally sits in the nucleus, but shuttles between the nucleus and cytoplasm. As motor neurons are becoming injured, there's depletion of that protein from the nucleus, and it aggregates into these skein-like or compact inclusions. It's prognostically heterogeneous, so the Americans sometimes call MND Lou Gehrig's disease. He was a fantastic Yankee Stadium baseball player who developed MND in his 30s. He had classical rapidly progressive MND, but Professor Hawking, of course, kept it at bay for nearly half a century, though he was very disabled in his latter years. And we know from years of studying it that the process of motor neuron injury, even if you know the upstream gene, is really complex. At least 11 different things go wrong: oxidative stress, excitotoxicity, mitochondrial dysfunction, etc., not only in the motor neurons themselves, but in the neighborhood cells. So the crosstalk between astrocytes and microglia, and the motor neuron also gets deranged. And this heterogeneity has hampered development of neuroprotective drugs. So we've had Riluzole, which has a very modest effect on life expectancy since 1995, and then nothing else until 2017, when edaravone came through, an antioxidant that is licensed in the US, but not in Europe or the UK. So it's a heterogeneous disease, and we need better ways of stratifying the patient so that the right treatment gets to the right patient at the right time. And I think we've got a lot to learn from oncology. So genetics can help with this start and subclassification. And I'll just show you some results from a systematic study that we've done called Ambrosia. That's a program, 3 centers, Sheffield, Oxford, and London, and we basically take biosamples from MND patients at the time of diagnosis, blood in all its forms, skin cells, CSF, urine, etc. And we also have the funding to be able to screen on a research basis all the known genes. So I won't go into the details of how we screen. You all know about Illumina technology, and we had standard methods for ascertaining whether a gene change was significant or not. And we just looked at the first 100 patients that went into Ambrosia from Sheffield. 7% of the 100 had a family history of disease, so would be predicted to have a pathogenic variant. But actually, when we systematically studied all of the known genes, 21% of that 100 had a clinically reportable pathogenic variant. Another 21% had a variant of uncertain significance after benign changes in healthy controls were removed. So if you look hard, you find a rogue gene in quite a lot of these patients. And this was a small-scale study, but we confirmed it in a larger study. So we have this big Project MINE European consortium doing whole genome sequencing on 10,000 MND patients. At this stage, it was more than 6,000 that had had screening, and they found exactly the same thing. So if you look for all the genes, you'll find one in about 21%. So we're trying to use this evidence to persuade Genome England and so on, that we should be at least offering genetic testing to all patients with MND, regardless of whether they've got a family history. So incorporating experimental medicine into clinical trials helps us dissect out subgroups, and I'll show you an example here. So some early work from Houston in the US showed that the higher the patient's regulatory T cell count, the better the prognosis. So people that naturally have high Treg lymphocytes have a slower disease course. So we embarked on this trial of low-dose interleukin-2, whose action is to increase Treg count and also Treg function. And we did a preliminary trial, just 3 months, 12 patients on high-dose IL-2, 12 patients on the lower dose, 12 patients on placebo, 5 subcutaneous injections per month in this cohort. And you can see in the higher dose cohort a very nice upregulation of Treg count, but also, importantly, a reduction in this inflammatory biomarker CCL2, also known as MCP1. And we took the PBMCs from this small-scale trial, and this was work done by a very talented young scientist, Ilaria. So we took PBMCs and did gene expression profiling. using a nanostring platform. Again, I won't go into all the technical details, but what we found is that in the treatment group, some patients got a high response in terms of their Treg levels. Others responded, but at a much lower level. And so we looked at the gene expression differences, and in a nutshell, what we found is that all the treated patients had a sharp downregulation of pro-inflammatory pathways, but the low responders, the people that got a less good Treg count, had a much more inflammatory phenotype at baseline. And we all know that in MND and other neurodegenerative diseases, neuroinflammation is an important component. So that preliminary trial allowed us to go into the MyraCals trial, funded by the EU Horizon 2020. Nigel Lee, it was the UK chief investigator. Gilbert Ben-Simon in Paris was the program lead. So 220 patients went into this trial. I'm going to just show you some headline results. So the biomarkers, the Treg cell count and the reduction in CCL2 confirmed the earlier trial findings. But what we also found was that neurofilament levels at baseline, and in this case it was phosphorylated neurofilament heavy, were really strong predictors of the survival. So if you had high neurofilament levels at baseline, your survival curve was much worse than people with lower neurofilament levels. And so this was a survival study at 18 months, and there was a pre-specified adjusted analysis taking into account these neurofilament levels. So the unadjusted analysis showed a 19% decrease in the risk of death at 18 months, but this did not reach statistical significance. But the adjusted analysis, taking into account the neurofilament levels, showed a decreased risk of death of 73%, which is quite significant, I think. So motor neurons are stuffed full of neurofilament proteins. As they die, they release those neurofilaments into the CSF and into plasma, into the blood. So using human cellular models is really important. So these days, we can take skin cells, take them over to the laboratory, bulk them up, and reprogram them to become motor neurons, other types of neurons, astrocytes, and so on. And we know a lot about iPS cells, where we take the cells right back to embryonic stages. We do that, but we also do a direct conversion of these fibroblasts using through induced neural progenitor cells. And the beauty of those is that they retain the features of aging, which is very important for neurodegenerative disorders. And they're also non-clonal and tripotent. You can make them into astrocytes, neurons, and oligodendrocytes. And we've showed in these cell models, reprogrammed cell models, that they recapitulate the pathological features of MND. So here is the TDP43 mislocalization. Here we can see misfolded SOD1 protein. And when we make human astrocytes, shown in red in this cartoon, and we grow healthy GFP lit up motor neurons on top of the astrocyte layer, the astrocytes from MND patients, whether they've got C9orf gene change, SOD1 gene change, or sporadic ALS, those astrocytes from MND patients are toxic to the motor neurons growing on top. And that allows us, that co-culture system allows us to screen drugs. And again, I won't go through all the detail of how we do it, but because the motor neurons are lit up with GFP, we can use an Opera Phoenix automated analysis system to assess motor neuron survival. And here we can take those co-cultures and try a range of different drugs. So we tried riluzole and edaravone that I've talked about, and a new drug that came from our preclinical work in our research institute. And you can see just the graph shows the level of motor neuron survival in different subgroups of MND patients and with the different drugs. And you can immediately see the heterogeneity of the response. And I'm very pleased to say that the drug that we pulled out was doing better than any of the other ones. And again, you can look at the transcriptional signature of responders, and in a nutshell, what the responders to treatment, so these are the non-responders that are localized with controls, and the responder treatment, the patients that responded best to that treatment got a much more significant change in their transcriptomic signature. And you can see this on heat maps. So here is the control group, the transcriptomic profile from astrocytes in the control group. These are the responders before treatment and after treatment. These are the non-responders. And you can immediately see the non-responders didn't do very much in relation to treatment, but the responder group converted their profile from the abnormal state at the beginning to much more like the control group following treatment. So I think transcriptomics is a very nice way of dissecting into responses and non-responders. So I'm just going to tell you now about one of the breakthroughs that has come in MND, and it's a genetic therapy for SOD1 MND, so the first gene that was found. So only 2% of patients, or 20% of those with familial disease that have a SOD1 gene change. This is intrathecal antisense oligonucleotide treatment called tofersen to lower the level of SOD1 in the cerebrospinal fluid and in the nervous system. And in the initial experimental medicine studies, first into man, so going very cautiously with a low dose and building up to a 100 milligram dose, the 100 milligram dose, a very nice lowering of the SOD1 level in CSF, so a good biomarker of target engagement. But really excitingly, what we also found in that highest dose group was a lowering of neurofilament levels, phosphorylated neurofilament heavy, neurofilament light, coming down very nicely in both plasma and CSF, suggesting, because motor neurons are releasing that as they get damaged and die, suggesting that we're protecting the motor neurons. That went in front of the FDA. They said do a phase 3 trial with that 100 milligram dose. And again, that was published a few months ago in the New England Journal of Medicine. And what was also shown in the phase 3 study, the treatment group in green, a very nice lowering of CSF SOD1, a huge lowering of plasma neurofilament light. So plasma levels are just as good as CSF. And the results are complicated. In a nutshell, the biochemical indices were there within 8 weeks or 3 months, but the clinical benefits took longer to show themselves. Not surprising when we're trying to heal motor neurons and get them to reinnervate muscles. So the primary endpoint was the change in a disability scale at 6 months, and that wasn't met, but when the patients were followed out to 12 months, the clinical benefits were obvious. And the FDA has, at the end of April, has given expedited approval for this treatment for SOD1 MND. NICE are considering it at the moment. goodness knows how expensive it's going to be. We'll have that battle to fight in due course. So the Tofferson trial really suggests that neurofilament levels might be a good biomarker of therapeutic efficacy. And we need to do more work, but if that is confirmed, there will be the possibility of shorter trials with biochemical signals of efficacy within 3 to 6 months, but also the possibility of picking out responder subgroups from this heterogeneous population. And also, I think what the 2 trials I've just briefly run through have shown is that stratifying patients at the beginning of a trial by severity of disease, rate of progression, and neurofilaments also look promising from that point of view. So I'm going to close now. What I hope I've covered just very quickly is that the heterogeneity of MND has hampered the development of neuroprotective therapies. We do need to adopt a precision medicine approach based on the molecular subclassification of the disease to get the right treatment to the right patient, learning lessons, I think, from oncology. We can identify the upstream genetic cause of MND quite commonly, and I think in what is called sporadic disease, genetics are also very important, but it may be more than one gene change that is needed to bring the disease out. So we need to change our approach to clinical trials, embed in experimental medicine and biomarker components, where the field is really excited that we seem to have biomarkers of therapeutic efficacy beginning to emerge. And I've touched on just using a range of model systems. Zebrafish, fruit fly, mice are, you can ask good questions and get good answers from those. But I think the ability to make the cells in the nervous system that get damaged from just a skin cell or a PBMC donation is really going to take the field forward. So thank you for your attention, and thank you for inviting me to the meeting.